So look, when this is up there and not grayed out, it means it's recording. So you don't have to ask me. Cool? Okay, so that's what that's up there for. Okay, so I generally record in the 10 o'clock class because I'm a little smarter. I probably gain 2 IQ points between 8:30 and 10 p.m. And 10 a.m. 10 p.m. I'm dumb again. Okay, so, but by the way, I'll go ahead and tell you that I, generally speaking, 8:30 a.m. classes have slightly lower class averages than any other class, except for like 3 p.m. So I told them that I wanted them to try to overturn that this semester. So you guys are in collaborative competition with one another. I wanted to give them a little extra motivation. It's only 2 points anyway, and it doesn't mean that people don't do well. It's just like it's a little hard to get going, right? It's a little hard, especially for you crazy people who don't drink coffee. How do you even do your lives? Okay, so we were looking at this last time, and so remind me before we get going any further. In order for something to be considered inside the body, what does it have to cross? Epithelial membrane, exactly. But then, of course, you're not finished when you cross an epithelial membrane. So the example that we used last time was basically trying to get glucose from the Snickers bar that I ate all the way into a cell. Let's make it a neuron this time, right? Because neurons are really important. Let's just go through that one more time. So basically, we're going to have a transport mechanism that occurs here, yeah? But it's still not where it needs to be, right? We're going to have another transport mechanism that occurs here, going from, for instance, the interstitial fluid to the blood plasma. This is across a capillary epithelium. Luckily, transport across capillary epithelia is not usually problematic because many of them are porous, fenestrated or sinusoid. Does that sound familiar? Fenestrated or sinusoid capillaries. And of course, the ones that are right by the digestive tract are pretty porous because they want to do a lot of absorption. So now we've got the glucose in the bloodstream. You with me? Okay. And what we're going to have to do now is we're going to have to get it back out. Let's say these are brain cells, neurons that we want to deliver it to. Yeah. But we might have an impediment here that we didn't have here. Does anybody know anything about the capillaries in the brain that could prevent an impediment to transport? What is special about the capillaries of the brain? I'm not saying they stop glucose transport, but they give you another consideration. What is special about the brain and transport in and out of the blood vessels of the brain? The blood-brain barrier, exactly. So the blood-brain barrier, the capillaries in the brain are not porous. They're really, really protective of the tissue. So you might have a different transport mechanism here, yeah, than you did here, simply because of all of those tight junctions. Does that sound familiar to you guys? So let's say you get it through the transport mechanisms of the brain. And by the way, they're actually carrier proteins on the blood capillaries that get glucose. But now, of course, we have to get it into the neurons. I know they don't look like neurons. Work with me. Okay, get them into the neurons, and I'm going to have to cross what structure in order to get them into the neurons? The cell membrane. And I'm going to have to worry again about transport mechanisms to get them into the cell membrane. And so we talked about this a little bit last time. In order to get something from the extracellular fluid to the intracellular fluid, you're going to cross the cell membrane. And so what we are going to gain this semester is good predictive ability about how you do that. How do you cross the cell membrane? And it's not magic, and it's not memorization. It has to do with the chemistry of the cell membrane and the size, charge, solubility of the molecule that you're trying to transport. With glucose is relatively large, like a mid-sized water-soluble molecule, so diffusion straight through the membrane is not an option. You need to use a carrier, yes? And we talked about this last time. You need to use a carrier into cells. And the vast majority of your cells to move glucose from the extracellular fluid to the intracellular fluid will use an activated carrier that is activated with a hormone. And what hormone is that? Insulin, yeah? It uses insulin to move glucose from the extracellular fluid to the intracellular fluid. With me? Okay. That just opens up the carrier in most cells and it moves in. So, of course, now we can bring up diabetes for one of the 3,000 times that we will this semester because it's such a fantastic physiological learning tool. And, of course, if you are dealing with human health, you are going to be dealing with people with diabetes. You just are. okay? So what is going to happen is with diabetes, you lack the insulin, can't open the door, the cell, neuron, cardiac muscle cell, whatever the hell you want to talk about, the cell will be swimming in food and starving. And so what we said last time was, what is the issue for that cell that has no glucose? What can it not do? Can't make any ATP over a decent period of time. If you can't make ATP, you die. And if it's an amitotic cell, like for instance, a cardiac muscle cell or a neuron, it's gone after it's gone. Which brings us to, of course, have you guys heard of diabetic neuropathy? Diabetic neuropathy is when untreated, improperly treated, whatever, diabetes will cause neurons to die. And as the neurons die, you lose both sensory and motor capacity. It's not usually CNS capacity. It's usually peripheral nervous system capacity. So does that make sense to you guys? Okay, so, and then we went through this. We talked about transport mechanisms, and we talked about the different kinds of body fluids, right? That's where we were last time, talking about the different kinds of body fluids. Realizing, of course, that there must be the capacity for transport between the body fluids, right? It's sequestered, but there is controlled transport between them. So now what we need to do is these are called body fluid compartments, by the way. And the only other thing that we didn't talk about last time is that, yes, your total body water is divided into the ICF and the ECF. And the ICF is, on average, 100 trillion individual pools in about a 150-pound person, 100 trillion individual pools of intracellular fluid, okay? If you are much taller or much bigger, you have more cells. That's how you get taller and bigger, with the exception of the fact that adipose cells can also get bigger than cells. But you also make more adipose cells when you gain weight. One of the most depressing things you'll ever learn in biology is that you can always make more adipose cells, but once you got them, do they go away? No, little bastards just stay around. Freaking house guests that don't take a hint. Unless, of course, you get liposuction, right? And then you can suck them out. But liposuction is this weird little thing that occurs. My best friend got liposuction, so I always like to tell her story because she's not here. She got liposuction because she didn't like her saddlebags like a lot of us, right? She got liposuction right here. But then she's just a normal human, and she likes a cocktail and a piece of pizza on occasion, so she started gaining weight again, nothing major. But since you don't have the cells here, she started gaining weight here, and she got herself a top butt instead of a butt down here. So she ended up with butt from here to here, and I don't know how that's better. That was a cautionary tale for me, is what that was. I was like, maybe I'll just live with what I got. So anyway, there is, however, a little teeny bit of ECF that is not plasma or interstitial fluid. It doesn't tend to be big picture physiologically important, but it exists. Little bits of fluid inside your joint cavities, because those aren't tissue fluid because they're not surrounded by cells. They don't have cells within them, right? Just inside your joint cavity. Or your aqueous and vitreous humor, those are extracellular fluid, but they're not plasma and they're not exactly interstitial fluid. So what the hell are they? They're that extra little stuff that you have. And those fluids are interesting. I mean, they're not particularly physiologically relevant most of the time, but because they don't turn over very quickly, so there's not a whole lot of transport in and out of them. And sometimes that's interesting. For instance, inside your eyeball, your aqueous and vitreous humor, it doesn't turn over. It actually takes years to turn over and get all new stuff in there. And the only reason that that's kind of cool is because when you die, it will narc on you. So if, for whatever reason, they needed to figure out what you had been ingesting in the decade before your death, whether it was poisons or entertaining substances, or they wanted to know something about your nutrient loads, you actually can get about a 10-year record out of your eyeball. I literally don't care. You guys can suck out my eyeballs and learn all my secrets once I'm dead, because I'm dead. And maybe it would make for an interesting discussion with the med school students, right? They'd be like, what the hell was her problem? I'd be like, I don't know, she had fun. Okay. All right. And then we got the intracellular fluid. We know, of course, that you are going to need to exchange substances between the ICF and the ECF across the cell membrane. And what you have known for quite a while now is many of the substances you exchange, right? Not going to surprise any of you guys that you move nutrients in quite often and wastes out. Nutrients meaning primarily large organic molecules like lipids and proteins and carbohydrates, those kinds of things, and waste products out, right? You move gases in, gases out, primarily oxygen in, carbon dioxide out, not news to anybody. But then one of the things that usually at this level people are less familiar with is the importance of moving ions, also called electrolytes, but in phys we call them ions. In nutrition they call them electrolytes, in and out of cells. Like you might know, for instance, about the sodium-potassium pump because you learned about it in 1610, but you might not know the why, like why we need sodium in higher concentration on one side than the other. You might know about the nervous system, but you might not know that we need calcium ions to cause synaptic transmission. You might not know that we need sodium ions to cause action potentials. So one of the things that you'll gain this semester that sort of augments what you already learned in biology is the importance of transport of these things, okay? So you'll know it pretty well by the end of the semester. Okay, so here's a discussion that we can have really quickly, is that, okay, so we of course know homeostasis. Before we get into any other questions, can anybody give me like a working definition of homeostasis? I'm not looking for a textbook definition. Working definition of homeostasis. Yeah, happy, regulated, balance. Homeostasis, homeo meaning the same, and stasis meaning stability. What homeostasis is a relatively constant internal environment. But constant doesn't mean constant, does it? So is my body temperature on 98.7 all through the day? No. So I think of homeostasis as kind of like predictable variability, which sounds like an oxymoron, but it's not. It means that if I looked at her or if I looked at him over the course of a day, I don't know what your body temperature is going to do, but I can predict the range that it's probably not going to leave. If you are a relatively healthy person over the course of a day, I would bet that your body temperature never hits 87 degrees Fahrenheit. I would also bet that it never hits 106, right? So the variability exists, but within a predictable range, yes? And hers isn't going to be exactly the same as his, but I would also predict the same thing about where he wouldn't hit. Does that make sense? So it's not constant. So they say relatively constant, but it's really not constant. It's like constant with an asterisk, right, associated with it. So before we get into the details of how that occurs, I want to talk about how costly it is to maintain a relatively constant internal environment. The metaphor I want to use is, okay, I need for some things to be true about the inside, the ICF, and some things to be true about the ECF, but I need for exchange to occur between them. But still they need to stay different from one another. It's like saying, okay, I need for the inside of my house to be 68 degrees, but outside it's 21 degrees. And I have 16 children running in and out, right? Is it not going to be expensive to maintain your house at 68 degrees when you have all of that transport? Does that make sense to you guys, that metaphor? Okay, so here's the deal. What your body does is it devotes quite a bit of ATP. Around 60% of the ATP that you use is just keeping you organized and homeostatic. But it devotes it to regulating the temperature, pressure, pH, concentration of one of these two, mostly, ICF or ECF. So which one do you think your body spends most of its money regulating, ICF or ECF? And I want to throw out to you that in this class, it does not matter if you get an answer wrong when we are discussing things, because that's how you get it right when you are being evaluated, right? Does that make sense? So there's no nastiness to one another. I won't be nasty to you. If it feels like I'm being nasty, it's because I think I'm being funny. But I usually only try that with people I know, right? So again, The question is, which one do you think you spend 60% of your ATP primarily monitoring, regulating, and fixing? ICF or ECF?
ECF.
He says ECF. Anybody say ICF? Okay, so there's arguments for both. But there's one thing that's true, and I can make it make sense to you. Okay, so you actually spend most of it monitoring your ECF. But let's think about that. Because how many pools of ECF do I have? One big one, right? That has like 2 at the most, right? And do they circulate? Yeah, the blood plasma circulates. So really the ECF is circulating and kind of coming into contact with at least most other body cells. So it's a circulating pool. How many pools of ICF do I have?
100 trillion.
100 trillion. Do they all communicate directly and easily to one another? No. So think about the cost of individually regulating 100 trillion pools. Now, I'm not saying the individual cell doesn't care about its own homeostasis. I'm talking about body-wide, where do you spend most of the money? You spend most of it actually monitoring, regulating the ECF. And here's another reason. I want you to imagine that I accidentally ingested a poison. Okay? I accidentally ingested a poison, and that poison could kill off this cell. Where do I want to assess it and launch a response? Do I want to wait to assess it and launch a response until the poison actually hits the intracellular fluid? Or do I want to detect it in the extracellular fluid, launch a response in the extracellular? Does that make sense to you guys? Okay, so it's more protective to spend quite a bit of your ATP assessing and monitoring the composition, pH, pressure, temperature of the extracellular fluid. It's more protective. It's also more energetically efficient to do so because, let's just hypothesize, whatever happens to the ECF if it doesn't get caught will happen to the ICF. And when you change the cell, you change the physiology. And when you change the physiology, you've got the possibility for a disease state. But if you can catch many things before they actually impact cell function, you can avoid the disease state. Does that make sense? Yeah? Okay, go ahead.
So is that kind of the idea? I've heard for a long time, of course we've had discussions about cancer, but generally it really flares maybe 3 to 5 years after you've had some major event that has dipped your body's ability
To respond
yes.
to it.
So is that part of that?
There are quite a few things that can depress your immune system. And when you depress your immune system, you are taking out quite a few of those checks that actually occur long before the cell activity actually occurs. So it's actually a pretty good explanation of it. Okay, so cancer is really complicated, but it's at least one of the factors that factors into cancer. Okay, so let us, so basically if the composition of the ECF changes dramatically, just a second, and you don't catch it, then you are predicting a future change in the composition of the ICF, which will cause a change in functionality. So Maria, did you have a question?
Yeah, mine's well, okay, have you seen that movie Blackwater?
Blackwater?
Yeah, it's really good. It's actually a documentary about like C8 and how it affects this whole town of
I haven't seen it.
in Michigan, and it caused cancers and tumors and it by this big company. So C8 apparently is it's a chemical in Teflon,
Okay.
like pans, and it said about 99% of people have it, and it can't get rid of it. Like you can't get rid of it.
Okay.
So how would that like go through your system?
I have no idea. It depends on whether it's lipid or water soluble and whether it's detoxified in your liver or your kidneys. And if it causes cancer, it probably impacts DNA function somehow. But I can't predict without actually knowing anything about the chemical. I mean, it's an interesting question, but we should look it up. Okay, who else had a question? Anybody? Okay, so now what we're going to talk about is okay, so we know that homeostasis is the goal. We also know that it's expensive, yes? Okay. So now the next thing that we are going to get into is the mechanisms of maintaining homeostasis. Yeah, mechanisms of maintaining homeostasis. And the primary mechanism of maintaining homeostasis is called negative feedback. Okay? And I know you've learned this term before, but we just need to really be able to recognize it when we see it in the future. So let's get a really good description of it and some analogies and things like that about it. So negative feedback. In this instance, what I want you to know is the term negative does not mean bad. Dump that definition of negative for this context, okay? It also doesn't mean lower than, okay? So with negative feedback, what you are going to do is to try to maintain homeostasis, which is a relatively constant internal environment. But let's narrow it to single variables so that we can think about it. You guys have probably used temperature quite often, but let's use blood pressure because I think you guys can think about blood pressure, okay? So what I can think about over the course of the day and my blood pressure is that I know it will go up and I know it will go down. But again, it's a predictable variability because I know that over the course of a healthy day, my blood pressure will never be 80/20. No, never going to be 260/150 over the course of a healthy day. Does that make sense to you guys? Okay. So I know that there's this range, right? And it's probably narrower than that, right? At which blood pressure is working. But negative feedback explains how you accomplish that, because negative feedback says that in this context, negative means opposition. It means if I want a relatively constant blood pressure, every time it goes up, I do what? Bring it back down. But every time it goes down, I bring it back up. So again, negative does not mean lower. It means opposition, as in you negate what's actually going on, okay? So it is bringing it back into the range. So negative means that any deviation from the normal value is resisted or negated, and the value is returned to normal. But what we need to think about as we go through this example is it actually returning to a set point or is it returning to a range? So think about that question as we're actually going through this. So I want to give you guys just an example of a negative feedback graph. So in Phys, FYI, you will see boatloads of graphs. And most of these graphs are going to be analyzing some variable over time. Yeah? Because you want to look over time to see how blood glucose varies, how body temperature varies, how blood pressure varies, how any hormone varies, anything, right? Look over time. And you guys know enough about graphs to know that graphs don't mean a damn thing if you haven't labeled the axes, correct? So with this particular one, you've got, it says time versus variable. I'm going to pick the variable and make it blood pressure, okay? Okay? So it is important whether the x or the y-axis is labeled properly. You've learned this before, but I'm going to reinforce it because people flip it, okay? The way this graph is set up says that blood pressure varies over time, or might vary. We're going to measure blood pressure over time, yes? But if you flip the x and the y-axes, you are telling me that time varies with your blood pressure, which is the lamest superpower ever. Shut up, Siri. Which is the lamest superpower ever. Does that make sense to you guys? Okay, so you can't flip the axes. Okay, of course, if this were a good graph, we of course have to label it, but we haven't got time for that. So what we're going to do is just do a hypothetical and look at what negative feedback looks like over time for blood pressure in a person. Okay, so you already kind of have it in your mind. You know, because you've been a person, that blood pressure is not going to be a straight line over the course of, let's say, 24 hours, okay? It's not, right? Your blood pressure is going to go up, it's going to go down, yeah? Okay, so let's predict what it's going to do. So what do you think your blood pressure is going to be if this were the range? We'll figure out the range in just a second, but if this were the range, what do you think your blood pressure was going to be when you woke up in the morning? Relatively low, yeah. It's relatively low blood pressure when you're asleep, right? Both because you're laying down and because your activity and also some hormones make it actually low, relatively low. But I'm going to make this person have the most kick-ass, accomplish everything day in the history of days, okay? They're going to accomplish every damn thing that they intended to. I swear I've had 2 of those days in my life. They were amazing. I remember them fondly. So here we go. This person is going to get up. And they are going to make themselves a very healthy smoothie, and they're going to have some coffee because they're not a monster, right? Smoothie and coffee. They cancel each other out. They don't. That's not Phys. Okay, But then because they are a person who's super admirable and is going to have a kick-ass day, they are going to work out in the morning. I don't even understand you people, but I admire you. We probably wouldn't get along. Work out in the morning. So, and then their kids are going to wake up. They got some kids. They're going to feed their kids, right? Then they're going to meditate. See, it's a good day. They're getting all the crap done, right? And then they are going to help their kids get dressed. Every parent in here knows that you can't find any shoes ever. So here, they've got 3 kids. Look for 1, 2, 3 shoes. Blood pressure goes up. Yes, you know? What the hell do they do with their shoes? I don't even know. And then they're going to drive the kids to work and to school, or to school and you to work. Then they're going to go to work. They hate their boss. That's the worst part of their day. Blood pressure goes up, right? They have their work best friend. Blood pressure goes down. Really, really hot person at work. You're not going to sleep with them, but your blood pressure is going to go up, right? Because you have a family and priorities. But don't tell me that doesn't occur. That's not even your fault. That's physiology. Okay. Go to a meeting, right? Get their day done, drive home, pick up the kids. Let's see what else you got to do. Got to make some dinner. They're going to work out again. What the hell? Come in, walk the dog, put the kids to bed. That's kind of stressful. Watch Netflix. Only they decided to watch You, the serial killer one, right? Blood pressure goes up. And then it's Friday. They're going to have sex, but it's Friday. There you go. Okay. So what we are going to see over time is that there is variability, yes, of course, right? And we got quite a few data points. But what we can see is we can see probably the ideal range for this particular person, because every single time you go out of the range, it starts to bring it back in. Does it mean the person never should go out of their ideal range if they are healthy? No, of course you go out of the range. Physiology and homeostasis says you bring it back in quickly, not that you never leave it. Does that make sense? Right? You're always going to go out of the range. And then, so that's maybe the top of the range, and then maybe the bottom of the range is like right here, because every time that occurs, they start bringing it back up. Does that make sense? Okay, so you can get a really, really good idea of this person's ideal range if you take quite a few data points. Do you think that, what do you think would be a better prediction of this person's range? 24 hours of data points or 72? Yeah, more data points is going to get you better information, right? Okay, and so the interesting thing about this is that until you get into things that are related to human health or maybe medicine, people don't memorize ranges, right? Because it gives them 2 numbers to memorize. They memorize means, yes? So you have probably one number memorized for blood pressure, 120/80, right? But that's not going to be useful for you as you actually get into clinical profession, whether it's nursing or PA or PT or whether you go to med school. It doesn't make any difference. Because you don't want to be the person who runs to the physician if the person's blood pressure is not 120/80, but 124/86. You're going to go get the doctor? Only if you want them to fire you. Then you're going to go get the doctor, right? Likewise, even if the person's blood pressure was here, do they necessarily need medical intervention, or should you probably ask them if they ran up the stairs? Right. So you don't stay in the healthy range all the time. What you do is you always go back into it. And the more quickly you go back into it, the more healthy your homeostatic negative feedback mechanisms are. Does that make sense? So right now, a lot of you guys are in your 20s. So in your 20s, your homeostatic control mechanisms, if everything else is good, are usually pretty fast. So if your heart rate goes up to, let's say, 145, give it a couple minutes and it'll be right back down, right? But is that true for an 85-year-old person who's running on a treadmill? No, their homeostatic control mechanisms are not as quick, right? And they stay out of range for longer. So what negative feedback graphs tend to look like over time is they tend to vary in and out of what you see that looks like a healthy range. Okay, so now So we've got two things. We've got homeostasis, which is the goal, yes? We've got negative feedback, which is the mechanism to accomplish the goal, right? Because it makes sense to you that if every time it went up, you brought it back down, and every time it went down, you brought it back up, you'd stay relatively constant. Now what we need to get is the actual parts of the body that are responsible for doing this. Does that make sense? And the parts of the body are collectively called a reflex arc that use negative feedback to accomplish homeostasis. So let's start with a metaphor first, and then we'll actually use a real human example. A metaphor for a reflex arc would be that in your home, right, if I set the thermostat for 68 degrees, right, it is not going to stay at 68 degrees all the time. It's going to go up, and then it'll be brought back down. It's going to go down, and you'll bring it back up. Does that make sense? Especially in like spring when the temperature is really variable. Okay, so what we are going to do is homeostasis is the goal. Negative feedback is the mechanism, but there are parts that are involved in accomplishing that. Okay, that makes sense? I'm going to define the parts for you. Okay, so a reflex arc has multiple individual parts. The components of the reflex arc. The first one is going to be that you cannot start all of this if you do not have a measurable variable, right? A variable that you can assess and detect change in. In our metaphor, the measurable variable is going to be room temperature, right? Okay, so I want to say that the room temperature that I'm measuring is higher than 68 degrees, right? Room temperature goes up. Everybody with me? Okay, so the room temperature goes up and it has to be measurable. But have we fixed anything just because we've measured it? No. So we have to have some other parts. The thing that measures the variable is called the sensory receptor. In this metaphor, the sensory receptor would be the thermometer portion of the thermostat, right? The portion that tells you what temperature it is. Yes? The thermometer portion of the thermostat is not the portion that makes the change, though, is it? Right? It just tells you what the temperature actually is. You guys with me? So the thermometer portion of the thermostat assesses. But then in order to make a change, you're going to have to send that measurement to the part of the system that knows what it should be. Okay? So now what I'm going to do is I just measured and they said it was 68 degrees. Let's make it 73. Let's make it 73 degrees, okay? Make it 73 degrees, and then I have to send that information to the thermostat. And the thermostat portion is the portion that has the ideal temperature set, the set point, right? The ideal. Okay, so this may be counterintuitive to you, but what it's set, you're going to have to send it through what we call an input or afferent pathway from the measurer to the brain, yes? And here it's a really short pathway because the wires are all together. In your body, the pathway is sometimes really long from the site of measurement to the site that actually knows what it should be, okay? So that's called the afferent pathway. And the thing that I send it to is called the integration center, the integration center. This is the brain of the operation for this reflex arc. This is the part that knows what it should be and, importantly, how much signal error is acceptable. Now, you may or may not know this, but it will make sense when I tell you. If you set your thermostat to 68, does your heater come on when it's 67.2? No, it doesn't. Does your air conditioner come on when it's 69? It does not. And why is that? Because it's incredibly inefficient to do that. So although we set it at a number, what we have is an acceptable range that we're cool with, okay? And you have that for body temperature and you have it for blood pressure. If you didn't, your heater and your AC would be coming on all the damn time and nobody can afford that, right? Does that make sense? So what happens is there's an error signal between the measured and the ideal. And what happens is the integration center assesses it and says, is that error signal big enough to do anything about? If it's no, then it just keeps measuring, right? The whole thing just keeps going. If it's yes, let's say it is 73 and you set it at 68, and they say that's entirely too much difference, then what it's going to do is continue with the reflex arc and send out a signal via the efferent or output pathway, yes, to something that can effect a change in that variable. These are called effector organs. Everybody with me? Okay. So what would be the effector organs for this metaphor in which the room temperature was set at 68, assessed at 75, the integration center said error signal's too big, send out the efferent pathway to where? To the AC, right? Right? Send it out to the AC. In that instance, the AC would be the effector organ, okay? But here's what's really important in physiology. The effector organ will effect the change, okay? It will change the variable, yes? So what in this instance it's going to do is that it's going to raise the temperature, right? In this instance, it's going to raise the temperature. But none of that is at all helpful unless you feed that variable back in and make a circle, right? So what do you do with the variable now that it's different? You feed it back in as the new stimulus. If you didn't, what would occur? It would just keep going, right? And you would have actually disturbed homeostasis in the other direction. Does that make sense? So you have to continue to do this over and over and over again. Okay, so logic question for you, and then you're going to do the Thinkpairshare, okay? So, and then we'll discuss the Thinkpairshare as well. So, some of these portions of the Reflex Arc are done almost continuously for your body variables, right? So some of them you just do all the damn time. Some of them you only do as needed. Anybody want to tell me which ones you should probably be doing damn near constantly? Which ones? Which of the A through G should you be doing all the time? You should always be assessing the stimulus and sending it to the sensory receptor, and then sending it via the afferent pathway to the integration center. Do you always have to send it out? No, because if the error signal was acceptable, right? If it was 68.2 and not 75, then you just said that's fine. Anybody want to try to hypothesize why you would spend the energy, because it all costs energy, on all of those first one, A through D, just sort of constantly? Or the other way to ask the question is, why do I not just assess my body temperature or my blood pressure or my blood glucose every 3 days?
Is it because, well, obviously because something can happen, but also because I feel like it would take less energy to do like a constant slow
Oh, that's
assessment?
good too. There's two reasons really, right? A hell of a lot can happen in a few hours, right, to your body. A lot can happen. If you're a parent, you know that a little kid's fever can spike from normal to 105 really, really quickly, right? And then in addition, if you let the problem get really big, it's way, way harder to change than if it's relatively small, right? So what's happening is you're constantly assessing the state. And this is really important to understand with, like, for instance, some kind of disorder. Like, why is it that if I had a thyroid disorder, for instance, and the thyroid regulates your metabolism, and you're constantly checking your levels of thyroid hormone to see whether your metabolism needs to go up or needs to go down, right? But if I had a normal thyroid, that's all going on real time, right? Feeding that information back in, assessing, figuring out whether something needs to go up or go down. Do you think that if I had a relatively normal, homeostatically, internally regulated thyroid, I would be more or less likely to have metabolism problems than someone who had to supplement thyroid hormone and have it checked every 2 weeks? The healthy person, the person, right, why? The person who has to supplement thyroid because maybe they had hypothyroid and then have the thyroid levels checked and their metabolism assessed every 2 weeks, can you see why they're way, way more likely to have metabolism problems, hypo and hyperthyroid? Does that make sense to you guys? Right? So the sort of take-home message to that is something that you already know but is worth verbalizing. There is no medication that is a substitute for a healthy physiology. There isn't one, right? It just doesn't actually feed into the system and work as well and constantly check itself as well as anything else. What was your question? Oh, okay. Okay, does that make sense to you guys? Okay, so what I want you guys to do is I want you to try a real world example in a Thinkpairshare. I want you to do it together. Again, with Thinkpairshare, I'm going to pop up the question. Generally, we're going to do them twice, but this first time we're just going to try it once. I'm going to pop up the question. You're going to take a couple minutes or maybe a minute to read it. And then you're going to discuss the answer until you come to a consensus with the other person. The important part here is not actually coming up with the right answer. It's being able to verbalize your logic of the right answer. If the other person has a better argument, you can change your position because that's learning too, isn't it? Okay, so you need to get at least a pair, but there can be 3. That's fine. Okay, so get ready. This one's not outrageously hard. Again, if you do not have Top Hat today, don't worry, still engage in the process. I'll give everybody credit, okay? One more reminder and then I'll shut up. You get credit for participation in Thinkpairshare questions. Participation is the learning, okay? Regardless of whether your answer is correct or incorrect, but I always will make sure that we understand the logic before you guys leave. Okay, so read the question and then I'll pop up the answers. Ready? Okay, and open book if you want it to be. It's fine. Okay, there's the answers. So if you were by yourself, you were doing it wrong.
But I feel like it's also
And Ruby, don't tell them.
Oh, you
well, yeah, it's probably my game right now.
You just got as much as you want, but don't spill. I'm gonna spill it. I know I'm ready.
See the objective though, the objective is positive change. It's talking about positive change.
Yeah, but so the response is the signal. I just said the functor is working.
If you are not talking to people, you are not earning your participation credit.
Oh, okay.
Responding to signal.
So the hardest one, because I was okay.
So I'm integration. Because I mean, that obviously can be sense. Or the Thinkpairshare pathway, obviously. Those are pathways. The heartbeat track.
Yeah, so you're saying the hardest one is
It's the integration.
yeah.
Yes.
First day one. The link for the lecture notes and then you have to register for your knowledge for the registration. It's not a paper. I only got college password. I'll bring some password. I have password. So
not send it.
Five more people. Three more people.
Does mental avocado is a process of change?
They're asking, what does the new avocado possessions or whatever?
One more person. 5, 4. Get your answer in. 3. You have to hit submit. 2, 1. The little airplane has to go. All right. Okay. So what do we think the answer is?
C.
We think it's C, Integration Center. 96% of you guys said C. But what I would like is to, A, tell you that C is correct. And B, I want somebody to explain the logic that they use to arrive at that answer. If you're comfortable. I'm not even calling you, make you do this stuff. If you're not comfortable, if you are, go ahead.
So the medulla oblongata is what was kind of like it received the information and also processed it, and then it sent it to the heart.
The receiver, the processor, and the sender, right, was the integration center. Okay, anybody use different logic? It's not the only way you can get there. No? Y'all use the same logic? In the last class they didn't, right? Yeah, so that's probably the logic that I would use, the one that both received the input pathway or afferent pathway and sent out the efferent pathway, right? And obviously it must be assessing to decide whether the efferent pathway is necessary. Does that make sense to you guys? So that's a human physiology example. It's not a particularly hard one. We don't have to start out with the hardest Thinkpairshare questions. We can start out with learning how they actually work. Do you want to do a hard, not one of these, but do you want to do a hard example of a reflex arc where the parts are a little stickier to try to figure out? Let's do diabetes. Diabetes is an interesting reflex arc. So with, well, actually, let's do blood glucose first, and then we'll figure out what diabetes has to do with this blood glucose. So the variable or the stimulus that we can measure in this particular instance is blood glucose, correct? Everybody with me? Okay. And so we need an afferent pathway. This time the afferent pathway is not going to go through the nervous system. Does anybody know how blood glucose travels? Travels through the bloodstream, right? It's going to go through the bloodstream. And then it's going to go to a sensory receptor. Does anybody know the primary place in the body that is actually measuring the levels of blood glucose? There's several places, but one of the primary places, this is why this one's kind of confusing for students. Where do you assess blood glucose? Measure? Yeah, so the pancreas gets all up in the business of blood glucose because it both measures the level, but it also sends it to itself via the input pathway that is also within the pancreas to the pancreas, which is the integration center for blood glucose, right? And the pancreas says, I want it to be this, it's this. Is that okay or not okay? Yeah? But let's say blood glucose was too high, higher than you wanted it to be. Then the pancreas would initiate an output pathway. But the output pathway, again, would not be into the nervous system. It would actually release something into the bloodstream. What would it? Insulin into the bloodstream to go to the effectors that could drop the blood glucose. Anybody know what some of the effectors are that can drop blood glucose?
The liver.
Well, the liver totally can. I didn't expect anybody to know that. Yes. And where else? What are the things that will suck up blood glucose when they meet insulin?
Cells.
Almost all cells in the body, exactly. So that is definitely an example, right, of both homeostasis, regulating the glucose level, yeah, Negative feedback, because it would go up and you brought it back down. And a reflex arc, only the parts get a little stickier when you're actually dealing with it. But you can actually think them through. Does that make sense to you guys? Everybody good with that one? Okay, so I won't make you do that one until we've actually really gotten into blood glucose. But they're not always as simple as you initially think that they are. Anybody have any questions? Got questions about this? You feel good about this question? Okay, so this is a large part of what we're going to be doing in phys. Is basically, here's a concept. Here's the application to the human body. I want you to critically think about the application to the human body and try to put it together. In the future, we'll start to break things and see if you can figure out what would happen, right? Like, you know, in any example, what would happen. So, all right. So next, what we are going to do is talk a little bit about some things that are related to this. And related to this is, so again, I want to put these three terms together because people get them convoluted and they mix them up. So I'm going to give you three terms and I want you to help figure out how they're related to one another. The term homeostasis, yeah, the term negative feedback, and the term reflex arc. Okay. Which one of these is the goal? Homeostasis. Which one of them is the mechanism? Negative feedback. Which one of them describes the parts? Reflex arc, right? So they're all related to one another, but you can't necessarily use them interchangeably. Does that make sense? Okay, so with that in mind, I want to tell you that your body is relatively smart, not intellectually smart in this particular instance, but what it can do is if you give it really similar experiences over time, what it can do is it can predict the necessary outcome without the actual stimulus. So let me give you a couple of examples. One of the things that will happen if, for instance, I eat a carbohydrate is that I will start to salivate to break down the carbohydrates. Have everybody with me? Then a whole bunch of other stuff will actually happen. But the stimulus was, of course, ingestion of the carbohydrate, and one of the responses was salivary gland secretion because amylase, salivary amylase, can break down carbohydrates. Everybody with that? So, but you've done that a lot, right? Right? You've eaten a lot of carbohydrates, and they're literally the best thing that exists, as far as I'm concerned. But sometimes you actually can have the response without the actual stimulus. So now let's say you walked into your grandmother's kitchen and you smelled chocolate chip cookies. Yeah? But you didn't get any chocolate chip cookies because she slapped your hand and told you that those were for Relief Society. Right? Okay? So what you would have is you would have the response that you thought was necessary, right? You're like, I'm going to get a jump on this because I know how this pattern goes, right? And what that's called is the feed forward mechanism, or this makes more sense to me. Feed forward is a little confusing to most people, but you do have to know it. Anticipatory homeostasis. Anticipatory homeostasis says, I think I know what's going to happen. Let me help you with that. Okay. Here's another example of anticipatory homeostasis. If you are a person who is very, very regular about your circadian rhythms, and you wake up every damn morning at 6:45, every single morning at 6:45, and for, of course, years and years, you set an alarm to wake up at 6:45, right? So you could wake up at 6:45 because you've got stuff to do. What will happen when you go on vacation and you don't set your alarm? You are going to wake up at 6:45 because you taught your body that your metabolism needs to start increasing because you've got stuff to do. You'll be more efficient at getting that stuff done. If you teach it over and over and over again that this is going to occur, it's just going to try to help you out and get you started because, of course, you're going to be more efficient if your metabolism is a little higher to get going. So it'll actually wake you up, right? Regardless of whether you want it to or not. Same thing happens in the evening if you go to bed every single night at 10:30 and you decide you're going to pull an all-nighter because you forgot that you're not 22 anymore, right? All-nighter sounds so unpleasant to me. I remember it used to sound fun. I was like, why would I do that? But what's going to happen when you decide you're going to hang out with the 22-year-olds and try to pull an all-nighter, right? You're going to fall asleep, exactly. So the same thing happens with lots and lots of things. If you're very, very regular about the whole stimulus response, then your body will go, hey, I think the stimulus is coming. Let me start the response. That'll be really efficient if it's right most of the time. If it were a person like me and I don't wake up at the same time or go to bed at the same time ever in my entire life, in part because my son is a morning person, I adore him. My husband is a night owl, I adore him. I always want to see both of them. I can't accomplish them both in the same day, right? So with me, my body would try to do that and fail so miserably that it wouldn't be worth the ATP to try anymore, right? It wouldn't be energetically efficient. So these are called feed-forward mechanisms. And please note that it's not the opposite of negative feedback. It's anticipatory, right? It thinks it's going to be right. If it's good at it, it'll keep doing it. If it's bad at it, it's not worth the money, right? Sexuality is another example. If you see your partner parading around looking very enticing, you may have an anticipatory homeostasis response. If, however, they immediately put on a coat and say they're going to a crazy party without you, the anticipatory homeostasis response was not useful. And can you dampen that over time? If it's always that kind of response, yes, you totally can. You can teach your body that that stimulus does not merit this response. That stimulus does not merit this response, right? If you like smelled grandma's cookies and she never gave you any, you're like, screw you, I'm not wasting ATP on you, mom, grandma. Okay, and then what we're going to do with the last little part instead of starting the types of negative feedback is that you guys are going to come up as you were leaving, and you're going to take just a minute or so. This chair is going to be set right here. I'm going to put on this video camera. All you're going to do, tell me your name as it's on the roll so I can find you. Tell me what you like to be called, and just tell me something about yourself so that I can learn your name. Why am I doing this? To torture the introverts? No, I'm an introvert. I'm doing this because if you are connected to the people in your class, including me, you are more likely to make it through the class, okay? So I'm doing it so that I can get to know you and I can call on you individually unless it stresses you out. I'll watch it, see if it does, right? But even when you raise your hand, I can say, thanks, Maria, for raising your hand, right? Instead of cold calling you. So here's what I want you to tell me. Name, what you'd like to be called, either something memorable about you, something important to you, or if you don't want me to know a damn thing about you, tell me a joke. Okay? All right. All right. If this doesn't go past there, I'll be able to see you. Okay?
Hey, Melanie, remember my name is Shannon? I used to teach biology, so I do.
Hi, my name's Ruby. You can call me Ruby or she got me at homeschool.
No. Well, I'm not the person who makes that decision, but she was saying it. The first time you go through this class, you have to take the lecture and but they are actually covered. I mean, you can ask, but it's not me. Oh, did you get that coloring look at?
She did.
Yes, so imagine should make it like some of the not all of it. I saw I don't know. All the this. What I would say Anybody know how much masculine is?
Hi, I'm Jamie. I like to go by Josie, and something interesting about me is that I guess it might not be interesting, but I have a cute little 6-month-old daughter and full-time mom too as well. My name is Grace, so I go by Grace. And something very interesting about me.